Okay, I wanted to take a little time to start off discussing a variety of what I thought were interesting and important things for you all to know. The first is really that while you're here because you're part of our community, NINDS is not the only institute at NIH which funds brain science. In fact, there are 8 institutes which are major funders, and some of you may have your disease or disorder represented not only in NINDS, but one of these other institutes. But it turns out that every institute at NIH funds some neuroscience. Now, the National Institutes of Diabetes, Digestive Diseases, and Kidney Disorders, NIDDK, actually funds neuroscience research because we all know that appetites in the brain, hunger, obesity is not only a fat cell problem, but it's also a brain problem. Cancer Institute, there are 2 major things they're interested in, brain tumors and also pain, cancer pain. So every institute funds. The major institutes are NINDS, and you're here, National Institute of Mental Health, National Institute of Drug Abuse, Aging Institute. About half of their funds actually goes to support neuroscience research. National Institute of Child Health and Human Development, developmental disorders and basic brain development biology, the Eye Institute, the Deafness Institute, and the National Institute of Alcoholism and Alcohol Abuse. Now, I've not listed, each of these diseases has its own mission, and I've not listed all of them, disease mission, but every single one of them funds basic science. And an interesting issue now is how do we decide, or how is it that enough basic science gets funded across all those institutes to keep a robust enterprise in that area? I haven't listed all the mission statements, but I would just refer you to the NIH mission statement, which is to seek fundamental knowledge about the nature and behavior of living systems and the application of that knowledge to enhance health, lengthen life, and reduce the burdens of illness. And this is an old statement because we took off the disability. Now, overall, NIH funds in 2012, $5.5 billion in neuroscience. Now, that's second only to the Cancer Institute and is a lot of resource. But I think when we all consider the extraordinary burden of brain disorders and brain diseases, you realize that maybe it isn't quite enough. Neurological disorders, we're interested in stroke, epilepsy, multiple sclerosis, amyotrophic lateral sclerosis, the muscular dystrophies, and a whole host of rare diseases. Epilepsies, maybe it's as many as 400 or 500 diseases. Psychiatric diseases, schizophrenia, depression, bipolar disorders, huge burden, drug abuse. Drug abuse is a brain disorder. Alcohol abuse is a brain disorder. Deafness, blindness. It's an extraordinary burden, not only in this country, but across the world. And so I think that the investment is maybe not even equal to the task of the burden. One of the most exciting things that's happened since we last met was this announcement by the White House, April 2nd, I think it was about 2 o'clock in the East Wing of the White House, and I was there along with 250 other people of the grand challenge, that is that the BRAIN Initiative. Brain research through advancing innovative neurotechnologies. It's kind of a catchy title. And the goal here is for several parts of the federal government, NIH, DARPA, NSF, and a number of foundations to work together to come up with, in the end, a really wonderful understanding of how the human brain works. And it was called the Next Great American Project. I'm not going to talk any more about this because Kathy Hudson, who is the one of the deputy directors of NIH, who has been responsible for leading the first parts of this initiative will be here to give us a talk in just a little bit. But I think the identification of brain science by the White House as the poster child for American innovation, creativity, investment in science reflects the excitement in the field, and you'll hear about that from Kathy, but also the extraordinary burden of brain disorders. And we all, I'm sure, have relatives, family, friends who suffer from, are afflicted by brain disorders. So this is a wonderful opportunity for not only the scientific community, but also for patients. This has been, in addition to the announcement of the Brain Initiative, a pretty amazing year for us. And I have to say, sometimes when you're down in the weeds dealing with the most recent dictum from HHS about who can travel where and that we can only give you water if you drink it from the fountain, we lose sight of how extraordinary the progress has been. And on this, I could have had 10 slides with print so small no one could see it. So I forced myself to come up with a list with print that maybe everybody could look at. And these are just a few of the highlights. So the Porter Neuroscience Building, which is on this campus and houses about half of the neuroscience investigators in the intramural program, will be dedicated and open in this October. About the time I became director, the first part of the Porter Building was built. And there was a plan for a much larger building. In the end, there will be close to 90 scientists all working on brain issues in this building. And they're going to be in the building not by a floor for NINDS and a floor for mental health and a floor for deafness, but put together in groups which reflect the questions they're asking. And we'd hoped to have the whole building done, second part right after the first part, but money got put into a different construction project. And when the stimulus funding became available, ERA, we had the plans shovel ready for the second half of Porter, and so we were able to bring the Porter building to closure. There'll be a wonderful scientific symposium on October 21st and 22nd, and then a dedication, and we hope that lots of people from Congress, congresspeople and staffers will come to that dedication, and it'll be an opportunity to learn about some of the wonderful things that brain sciences are doing. A second wonderful accomplishment is that NeuroNext is actually up and running. I think we've talked about that with you before. It's a wonderful new phase 2 clinical trials network, which has a common IRB. Doesn't take 70 IRBs to approve a project. It's one IRB and pre-negotiated site contracts. So we can go from protocol to patient in a third or a quarter of the time it used to take us. I think this is a spectacular accomplishment, and Petra Kaufman, who's responsible with her staff for doing this, is here. So the 3 projects include a spinal muscular atrophy biomarkers project in the immediate postnatal period of development, a project using an inflammatory drug, Abudilast, for secondary and primary progressive MS. And that is, interestingly enough, supported not only by NIH, but has a public-private partnership. So the National MS Society is helping with that, as is Medici Nova. And then the third one, which is about to start, is a phase 2 trial of rituximab in myasthenia gravis. This is looking at something that's been successful in other autoimmune diseases and might be of benefit for myasthenia gravis. So very exciting. And because that project has been as helpful and useful for the Institute as it has, we've just cloned it, sort of, in that we're going to establish this year and the beginning of next year a very similar network for stroke. Stroke is the 4th leading cause of death and disability in this country. It's in our name. While we have some treatments for acute stroke, we're not good at preventing, we're not good at salvaging brain after a stroke, and we don't know as much as we should about rehab. We have had a number of trials. In each trial, we've built up a network, run the trial, broken down the network, started a new trial, built up a network, run the trial, broken down the network, and so we are going to have a network which will do phase 2 and phase 3 studies in stroke prevention, treatment, and rehabilitation. And the first of those grants for the Clinical Coordinating Center and the spokes, the hubs of the clinical sites will go out before the end of this fiscal year. We had 2 clinical trials that finished this year. We had other clinical trials that finished, but I want to mention these 2, Aruba and IMS-3, because the results of these clinical trials, we believe, should influence clinical practice. And the first of these was Aruba. This was looking at interventions to affect small, unruptured AVMs, arteriovenous malformations. And so there's been this big discussion in the field. Do you do watchful waiting, or do you actually intervene? The trial was supposed to have over 500 patients. It was hard to recruit. In the end, there were, I think, slightly more than 200 patients. And even with only 200 patients, we got a very clear answer. The patients who had the intervention did much worse. So this suggests very strongly that watchful waiting is better. We will continue to follow those patients who were in the trial to see if, in fact, over 3 years, 5 years, 10 years, that remains true. And the second trial, so important for patients to know, the data in a randomized, blinded, phase 3 trial made it clear that there's not benefit right now. The second trial that we completed was looking at mechanical removal of clots in stroke, in patients with ischemic stroke where treatment with TPA was insufficient to get blood flowing again. A lot of belief in the community that this mechanical thrombectomy was of significant benefit, but when we did a head-to-head comparison, in fact, that was not the case, and there are lots of issues that need to be resolved about how the trial was designed, but right now the evidence does not suggest that. Induced pluripotent stem cells. So it turns out that you can take a piece of skin from an adult, a baby, a person with a disease, and put those cells from the skin piece into culture, and by magic, sort of, treat them with several factors which actually tell those skin cells, you're not a skin cell anymore, you're actually a pluripotent stem cell, which can turn into all the different cells of the body. This was an extraordinary finding by a Japanese investigator and 2 American investigators, for which Yamanaka and someone else who worked on frog dedifferentiation, Gurdon, thank you, won the Nobel Prize. And what you can do with these cells is you can take from Parkinson's patients or epilepsy patients or patients with schizophrenia or patients with MS or ALS or any disease and make these stem cells. And you can grow the stem cells in culture, you can turn them into nerve cells, and you can understand how the genetic mutation which caused the disease causes the neurons or the glial cells to misfunction. And it means you can learn a huge amount about how diseases are caused, how our diseases are caused. And before this, you either had to work in mice, which are good models, but not human cells. Mice, we can cure lots of disease in mice that when you take that treatment to a human, it doesn't work. And without trying to take brain biopsies or work on dead brain, just real human cells with the disease turned into neurons and studied. And the other thing that's incredibly exciting is that people are now using induced pluripotent stem cells, which have been turned into dopamine neurons, for example, from Parkinson's patients, to actually test potential therapeutics. And there are already beginning to be some very promising thoughts about how we would do drug development better. So you see I have this list. Another example, so earlier this year, we discovered 6 new epilepsy genes. Actually, it may be as many as 20, but 6 for sure. And we did this using next-generation sequencing. That's getting the sequence of all the coding regions in a patient with epilepsy, and then looking and seeing, okay, where are the differences from a normal person, and identifying where the changes are that cause the epilepsy to occur. And it was a very special population of patients because the parents of these patients were perfectly normal. So we could compare the parents' exomes with the child's exome and know that the differences between the two were responsible. And it turns out from this there are particular pathways that emerged as potentially causing the epilepsy. And an exciting way that this can be expanded is at 6 or 8 different centers in this country now, kids with epilepsy, which is unexplained, get their exome sequenced. So health insurance companies or parents are paying for the sequencing, and we're hoping to actually set up a big database that would collect not only what we're sequencing with your hard-earned tax dollars, but also what are being sequenced in essence for free by the clinical care. And we think that this is important for epilepsy, and there are lots of projects now in our institute and other institutes looking at how we can harness this next generation sequencing to other diseases. And then a final piece on my advances list is the release of the first 120 Connectomes. These are maps of the wiring in the human brain. This is a project that the Blueprint has been funding, and so there are now available for any scientist to use once they register maps of the connections from 120 normal young adults to look at connectivity, function. It's a pretty spectacular resource. And this is just one of the pictures from this project. It's looking at the myelin, the parts of the brain where the myelin is densest, and you can see that bright red strip over in the top 2 pictures, that's over the motor cortex. So this project eventually will have 1,200 connectomes, and it will be mined by thousands of scientists, and there are already beginning to be very interesting findings like if you have identical twins, some parts of their wiring diagram are identical, and other parts, those parts which are influenced by experience, can actually be different. So lots of exciting stuff going on. I wouldn't have done my job if I didn't talk about the budget. That was all the good news. There is spectacularly wonderful stuff. But this is the not-so-good news for the NIH, and I believe for patients and families of patients. If you look at this graph, you can see in about 2003, that was when I became institute director 10 years ago, it was the end of the doubling of the NIH budget and the beginning of the flattening. Now, I said not flattening of the NINDS extramural grants budget, but actually the undoubling, because in the green line, you can see that if you actually translate that into dollars, adjusted to 1995 dollars, we've actually lost a significant amount of purchasing power. And our purchasing power now is not really different than it was in 2000. And just think about how much has happened in terms of opportunities and discoveries since 2000 that we really aren't able to take full advantage of. Now, this is our particular picture for 2013. So the sequester got put in place. All discretionary funding got cut 5%. At NIH, because we're in the Department of Health and Human Services, we also had a transfer of 0.7% of our budget out of NIH institutes and into the department to enable the department to actually put in place the Affordable Care Act. Now, each institute was allowed to develop a plan that balanced cuts, the 5%, 5.7% that we lost from our budget, balance that against the different parts of the budget. So there's the money that goes out, the extramural grants that go out to investigators across the country. There's a portion of our budget, and that's about 87% of our budget. There's a portion of our budget, about 10%, that funds research in the intramural program on this campus. And some of you I know have had interactions with the intramural program. And then the other last piece is RMS, and that's what pays for all the staff who are program staff, my salary, Walter's salary, Marion's salary, and the grants management people. So what we did was we just cut everybody 3.5%. Now, you're thinking, okay, they got cut 5.7, but they're only cutting 3.5. Is she going to go to jail at the end of the year because she's got a shortfall? In fact, we were able to manage this year to only cut, can I believe I'm saying that, only cut 3.5% because we'd had some really big grants that had ended, so that those funds were available. We have been unbelievably diligent in looking at all of our legacy programs. During the doubling and right after, I have to say the Institute was a little like a kid in a candy shop, and we created all kinds of totally wonderful, cool programs, which some of which worked, and accomplished their mission, some of which didn't work, and some of which probably were never going to get to the goal that we wanted them to, and in some cases, the goal wasn't as important. So we only had to cut 3.5%. So, and efficiencies in clinical trials. I don't want to leave this off because Petra has just done a spectacular job of managing our clinical trials, infinitely better than we did before. So what are the consequences of this? It's hard to say, science cures come to an end, we'll never have another treatment for a neurological disorder. Because we've managed not so badly this year, and I think many investigators have managed okay. But it's very clear we've cut ongoing projects, and I bet for those of you who fund research that those grantees came to you and said, oh my God, NIH has cut my budget, can you help me out? Or for those of you who fund grants, you've got lots more grant applications this year than you have in the past. There are new projects which weren't funded, and we will never know what those projects would have given to our understanding of disease and therapies for disease. In the projects that were funded, the progress is going to be slower and opportunities will be lost. Now, the question is what's going to happen this coming year? And to be perfectly honest, we really don't know. If the Congress actually is able to create a continuing resolution, which we're all optimistic will happen, then we will end up with the budget for 2014 that we had in 2013, except the full sequester wasn't levied on us in FY13 because they managed to find $24 billion that they counted against the sequester in 2013. So we will have not only the current 5% cut that we had, plus an additional 2% because they don't have that money to make up. We think the continuing resolution will last until December 15th. That's what I've read in the Washington Post, but then we simply don't know what will happen. And I am absolutely convinced that the consequences of these cuts will be cumulative, and that all the tools that the Institute and investigators use to manage this year won't be available next year, and we will see really significant detriment to our mission, and our mission, I know, is your mission, better treatments for patients. So I have, I think, 5 minutes left, and I would love to take a few questions if anybody has them. I apologize for the whirlwind tour of our advances and the very dismal outlook for the future. Yeah, Susan.
I wonder what your thoughts are on, I'm sorry, your thoughts on the impact of sequestration and the cuts on the next generation of scientists.
So if you had a kid, if you had a child who is graduating from college and interested in science, I don't think there's a single person who works at NIH, who works at an academic medical center, who works on diseases, who works on basic biology, who would advise that child, that young adult, to go into this field. I know when my son went to school, he was class of 96 from high school, so 2000 from college. We thought, absolutely, think about a career in biomedical research, and I would never recommend that now. So the best and brightest who could have made extraordinary contributions to this area are looking very carefully at whether or not this is something they, and even the ones who are doing it are saying, well, I really love being in the lab, I really love making discovery, and so I'll just do this and get my PhD, but then I'll think of something else to do. And PhD training is wonderful training. You learn how to solve problems and to think creatively and to look at the literature. And physicians, why, if you were a physician, would you want to go, why would you want to think about research? And for people who are doing disease-related research, physician scientists are absolutely critical. So there's talk about losing a generation, and I know it always sounds like hyperbole, but I think it's really, really true. So more dismal news. Yeah, way up there. Right. So the question is, given that I really hyped induced pluripotent stem cells, and they're a wonderful tool, what is the Institute or NIH doing to create repositories for either fibroblasts that you would turn into these cells or the cells themselves? And NINDS, with our stimulus money, God bless we had that money, long gone, actually picked three of our diseases, ALS, Parkinson's disease, and Huntington's disease, and put money into consortia which collected the fibroblasts, created induced pluripotent stem cells, and we have put them into a repository. And we got funding, once the ERA money ran out, we got funding through public-private partnerships with companies to continue to work on those particular cells. So we have the capability of doing it. Now, if we have 300 or 400 or 500 diseases, now not all of them you'd want to have induced pluripotent stem cells from, and it costs X tens of thousands of dollars per disease, then we might end up only collecting induced pluripotent stem cells. But we are certainly open to the idea of a partnership with individual foundations to see if we could work with you to take advantage of the fact that we have a contract for this repository. Okay. Any other questions? Yeah.
What percentage of the funding of NINDS goes to exploring infectious etiology, and does the NIAID spend any money other than, is there any non-HIV money spent on infectious etiology of CNS disease?
So we get, we have a small amount, we have a small portfolio of grants that looks at infectious etiology. It's handled by Mei Wong, who also handles the HIV portfolio. We work very closely with NIAID to make sure that, to the extent possible, we're funding as broad as many of these kinds of projects as we can and not duplicating. Also, I would say that in the intramural program, the clinical director, Avi Nath, actually is very interested in infectious and immune consequences of infectious disease and the roles that it plays. You know, it's interesting, 10% of the NIH budget gets spent on HIV, and a recent JAMA article looking at burden took HIV from probably in the top 10. I don't remember exactly where. I think it's down to 27. So one of the complicated issues is, as we look across the whole NIH portfolio, how do we determine allocations like that? But that is not, I mean, that's a legislative question and way above my pay grade. Last question, because I don't want to steal Petra's time. Yeah.
Okay, well, this is a good question because it's looking for a ray of hope. You talked briefly about the BRAIN initiative,
Right.
and how that's the next great American project.
Right.
How does that provide a bright spot in terms of the cuts and the future of the science and the discoveries, particularly for the group that's here today in your institute?
Right. So there's an initial commitment of $40 million from NIH. There's also commitments from other federal agencies to jumpstart this initiative. And on Monday, the planning committee that has been putting together the NIH, the proposal for NIH to consider, will be reporting out their first recommendations. They'll have a final report in June. Half of that money is money that is allocated towards large initiatives like the Human Connectome project that I showed you, as well as some money from Francis Collins. And the hope, I would say the hope, is that as this project gains momentum and there's recognition of the really important contributions that it can make to our understanding of what makes us us, the human brain, and how it works, and to our understanding of disease, that money will follow. But you know, if I weren't an optimist, I wouldn't still be in this job. So I believe that there will in fact be an increasing investment. And in terms of the immediate applications to disease, which I think is something we certainly worry about, and I know many of you do, the kinds of things that I can see happening absolutely right away are changes in the way we think about deep brain stimulation. So for Parkinson's disease, in the mid stage of the disease, after L-DOPA no longer works or dopamine agonists don't work so well, you get stimulating electrodes implanted bilaterally in one of 2 or 3 different places, and they're a little like a telephone pole. They work, but they're certainly crude. They don't take advantage of all the understanding that we have of circuits and the understanding we will have. And imagine, we talked to someone recently from OMB who came out to visit NIH, and she knew someone who had recently gotten DBS, and it certainly helped with the walking, but had done nothing for the wooden face. And so an obvious hope would be that if you knew more about circuits and had a better way to stimulate, you could actually treat all the different motor manifestations and some of the cognitive manifestations. Also real implications for epilepsy.